Hello everyone, we are from Group 4. My name is Aman and these are my friends, Hanis, Aini and Siti. We would like to present to you about age estimation of DNA from forensic DNA fingerprinting to forensic epigenomics. Let's get started. What is DNA fingerprint? It is a forensic tool that links evidence and a suspect in criminal investigation. Now, let us see the procedures of DNA fingerprinting. First of all, blood sample is collected from a crime scene. Then, the DNA is extracted from the blood using specific procedures. Next, the extracted DNA is cut into small fragments by using restriction enzymes. The extracted DNA is then separated using gel electrophoresis to form many fragments. After the separated DNA fragments are incubated and cooked, they are viewed under X-ray. The further the DNA fragment in the gel electrophoresis, the smaller the size of the DNA. Due to some disadvantages, this technique is later replaced by short tandem repeat analysis. Short tandem repeat, also referred to as microsatellites or simple sequence repeat, are accordion-like DNA structures containing 4 repeat units in length between 2 and 7 nucleotides that are tandem repeated from around half a dozen to 12 times. It has become the gold standard of genetic identification due to advancements of technology that is capable of amplifying repeat STR loci named multiplex STR. Now let's move on to the function of DNA fingerprint. The function of DNA fingerprint is for criminal investigation, which it matches DNA obtained from a crime scene to the suspect DNA sample obtained from DNA database. Now, let us see what are the differences between RFLP and STR analysis. For RFLP, it requires restriction enzyme to cut DNA into fragments, while STR analysis does not require restriction enzyme. Next, RFLP does not require PCR to amplify the DNA sequence, while in STR analysis, it requires PCR to amplify targeted DNA sequence. And lastly, RFLP needs a large amount of high-quality DNA, while in STR analysis needs only a small amount of DNA.
I'm Hanis. I'm going to talk about the limitations of confirmatory forensic DNA testing. STR profiles can help in investigations only when the perpetrator's genetic background is available for analysis. National DNA database may provide new evidence leads in cases without perpetrators. However, they are not necessarily usable due to a lack of connections. In order to overcome this, the suitability of STR genotypes to infer the donor's geographic ancestry has been tested in an effort to provide forensic leads to non-matching STR profiles. At first, an online calculator called Pop Affiliator was developed by Pereira et al. 2011 and is able to identify the STR profiles from 3 main population classes of East Asians, Europeans, and Sub-Saharan African. Further development with more precise estimation of the geographical history of an unknown DNA sample was made by examining ancestral informative SNP markers, whose bi- or tri-allelic distributions across populations are representative of continental-scale geographical backgrounds. For instance, the advancement of massively parallel sequencing techniques in forensic have broadened the ancestral informative SNP markers PCR multiplex, which can be used to distinguish between 5 major subcontinental populations: Europe, East Asia, Native America, Oceania, and Africa. Other than that, later study explores the use of microhaplotypes, mitochondrial DNA, and Y chromosomes to infer the maternal or paternal ancestry of a DNA sample. Besides, externally visible characteristics, or also known as forensic DNA phenotyping, targets to analyze DNA extracted from unknown stains in the crime scene to provide proof of stain donor appearance prediction. Pigmentation-related ABCs is one of the successful applications of forensic genetic markers to predict the hair, skin, and eye color.
Hi, my name is Siti and I would like to explain about markers and methods to estimate human chronological age. Chronological age plays an important part in criminal investigation where it will help in human identification. Previous studies stated that analysis of human skeletal system become one of the important methods, but it has limitation where it's only relevant to solid tissue. The main advantage of molecular genetic method is they can be applied to any tissue that contain DNA where a source of DNA fingerprinting can be found. Generally, the amount of nucleic acid in forensic specimen is not enough for identifying DNA. Two methods has been developed, which are targeted mRNA and CpG site assay, specifically for small amount of DNA to estimate age with marker. However, for established techniques such as pyrosequence, snapshot, and epityper are used for large amount of DNA, range between 200 nanogram to 5 microgram. Different types of CpG markers can be found in saliva, blood, teeth, and semen. As the result for methylation patterns, 7 highest age-related loci are found and can be accessed through online tools.
Hi, this is Ainin. I will be explaining on the current and future insight of forensic analysis. Methylation-based analysis is one of the current promising techniques for estimating chronological age in forensic analysis. European Union's Horizon 2020 Research and Innovation Program is one of the few programs that is involved in developing analysis for estimations of chronological age. They are responsible for finding the best methylation markers and optimizing standard protocol and method of the analysis. However, there are some limitations and challenges in developing the most suitable methylation marker. Firstly, different tissues demonstrate different methylation patterns. Next, stabilization of methylation marker over a longer period of time are in question. Next, the emanation process on ancient sample could give inaccurate result of analysis, effect of sex on the sample analysis, and lastly, limited dataset on variability of methylated base age geographically. Now, let's look into some current findings. According to Cho et al. 2017, investigation of CpG markers are tested in Polish population samples by using a dataset of Koreans. They found out that there is relatively consistent age prediction when using earlier developed model, but some markers are required adjustment with using new datasets. Finally, we have come to the end of the video. So here is the summary of the method development in estimating chronological age in forensic field, from RFLP to STR, and now the current method is methylation-based analysis.